Hi everyone, it's Dr. LeBlanc with a lecture about prokaryotic regulation of gene expression by microRNAs, riboswitches, and some post-translational regulation. Our learning goals for today are to understand how prokaryotic gene expression can be regulated by antisense microRNAs at the translational level, to understand how riboswitches or aptamers and small effector molecules combine to regulate gene expression at the transcriptional and translational level, and to understand how feedback inhibition modulates the express trait associated with a gene. This diagram shows a RNA for an OMP-F protein hydrogen bonded to a antisense small RNA. Now remember, this is prokaryotic gene expression. MicroRNAs are antisense small RNAs, that's this gray one. They can be produced quickly in the cell in response to environmental stimulus, and they can regulate gene expression very quickly by blocking translation of a particular messenger RNA. This particular messenger RNA encodes the OMP-F protein, which is important in osmoregulation in the bacteria. It's usually produced at low osmolarity conditions, and so if the cell suddenly encounters a difference in osmolarity, then you want to shut this protein off as quickly as possible, and that's what this antisense RNA does. This antisense binds to the messenger RNA and blocks translation. When we study eukaryotic gene expression, we're going to see other types of antisense small RNAs affecting translation in the cell as well. Okay, what happens if you have all your enzymes that you need to make a product, but you're making plenty of the product and you don't want to make any more, and you don't want to degrade all these enzymes that you just spent a lot of time making? Well, you can have your pathway be regulated by post-translational feedback inhibition. Enzyme number one in this pathway is an allosteric enzyme. Allosteric means that it has an other site, two sites. It has a catalytic site right there, which binds the substrate and then converts it into product. And it also has a regulatory site at a second spot in the enzyme. That second spot binds the final product of the pathway. When the concentration of the final product becomes high, it will bind to enzyme number one. There's our final product, and it binds there. When it binds, the enzyme undergoes a conformational change, affecting the active site of the enzyme, moving those amino acids in that pocket in a different orientation so that it can no longer catalyze the reaction. So this process is called feedback inhibition. Riboswitches. What are riboswitches? Riboswitches are alternate conformations of stem loops and three-dimensional structures of RNA, usually as part of a messenger RNA. They're also called aptamers. They can be a form of control for transcription and translation, also for RNA stability and splicing in eukaryotes, but we're only going to talk about transcription and translation control in prokaryotes right today. The switch from one active conformation to another inactive conformation relies on the binding of a small molecule to a pocket formed by the riboswitch RNA. That's what this is showing you a demonstration of down here. Here's a riboswitch. It is a part of the RNA. Here's a stem loop here and another stem loop here. The difference between attenuation and riboswitches is that riboswitches actually bind ligands. Okay, and in this conformation, the terminator is formed, and you detach from the DNA, and RNA polymerase is no longer transcribing this message. We're going to look at the same pathway in two different bacteria, B. subtillus and E. coli. The thymine pyrophosphate or B1 vitamin pathway enzyme, or the thioperon. In B. subtillus, an aptamer forms to regulate gene expression at the transcriptional level, so we're probably going to be forming a terminator. In E. coli, we see regulation at the translational level. So in B. subtillus, the transcription terminator riboswitch looks like this. It has, towards the 5' end, it has several stem loops, one of which is an anti-terminator. If the TPP levels are low, thymine pyrophosphate levels are low, you want to make sure you transcribe all the enzymes to make more. So you end up with the formation of an anti-terminator, and transcription proceeds through the operon, and you make all the enzymes to make more TTP. What if you have enough TTP? Well, it turns out that the stem loops here in this riboswitch portion of the messenger RNA form a pocket to which TTP binds. When TPP binds, it stabilizes the structure, and you end up with a terminator stem loop with a bunch of U's following it, which terminates transcription, and you preserve your energy and don't make more of the enzymes to make TPP because you have plenty of it. So here we see a terminator, stem loop terminator here. What about in E. coli? Well, we have a similar looking aptamer, but the function is different. Instead, at low TPP levels, we have our Scheindel-Garneau sequence exposed, free to bind to the small ribosomal RNA subunit and position the start codon in the right place and have translation proceed. Remember, we are looking at a translation terminator here. However, if TPP is high, these stem loops again form a pocket, bind TPP, and end up sequestering the Scheindel-Garneau sequence. If the Scheindel-Garneau sequence can't bind to the ribosome, then the AUG won't be positioned correctly, and we won't be able to initiate translation. So there's lots of different types of riboswitches for transcription, translation, some for RNA stability, some for splicing. Of course, those are going to be in eukaryotes. And you can read more about it in your text or on the net. So in summary, prokaryotic microRNAs are antisense small RNAs that can bind to messenger RNAs and block translation. Feedback inhibition occurs when the end product of a pathway inhibits the first enzyme in the pathway by binding to an allosteric site. The conformational change that happens when the product binds to that allosteric enzyme inactivates the catalytic site. Riboswitches, or aptamers, are portions of RNA transcripts that can have three-dimensional structures formed from stem loops and bind small effector molecules. The conformational changes when they bind the small molecules affect a change in regulation. At the transcriptional level, riboswitches can act by the formation of termination stem loops, transcriptional termination stem loops. At the translational level, riboswitches can act by sequestering or releasing the Scheindel-Garneau sequence. And in eukaryotes, riboswitches also have roles in messenger RNA splicing and stability.